others, hosted by the Penn Museum, and I want to give special thanks to them, particularly Janet Monge, who has been fantastic to work with, and I hope we'll find other joint projects in the future. But this is one of those wonderful Penn interdisciplinary collaborations. Okay, so today's talk is focused to scams. What is this talk about? Let me just begin preface these talks by asking that question, and then by answering it for you, and telling you what it's not about. This talk, this mini symposium, as we're calling it, is actually not about understanding racial differences in terms of brain size or function. This is not a talk on anthropology, physical anthropology, or neuroscience. What it is a talk about the way these sciences work, what they can and can't tell us about certain questions, and about the social context in which these sciences operate. And I think the easiest way to explain sort of what aspects of the social context we're talking about here is to refer to, by the way, I don't need any outside competition. Ratings of pain, when they go down, show that placebos reduce behavior shows the same as reduced pain. What's special about seeing less pain activation on the brain scan? There may be some ways in which it is relevant, but basically it's treated as a kind of evidence that trumps all other evidence. Oh, it's in the brain, it's real, it's essential, it's, definitive. So basically, science aspires to certainty, but there's nothing intrinsically more certain about brain science than there is about other kinds of sciences. And brain science, like all other kinds of sciences, takes place in a social context. And when scientists interpret their data, they do it within a theoretical framework that often has many implicit, unarticulated assumptions, including when the phenomena being studied are complex social phenomena, assumptions about humanity, societies, etc. And I think this is what we're going to see today in the following presentations. So speaking of those following presentations, let me briefly introduce our speakers. We'll first hear from Janet Monge. Janet, as you know, is a curator here at the Penn Museum and a faculty member in the Penn Department of Anthropology. And she kind of broke, was it the Morton Gate or School Gate? Yeah, right. Gould Gate, yeah, it's actually Gould Gate. So she will begin with this presentation. We'll then hear from Geoffrey Aguirre. He is a neurologist and neuroscientist, member of the Neurology Department, Center for Cognitive Neuroscience, and Center for Neuroscience and Society. And he will talk about the ways in which similar issues of measurement and interpretation arise today in studies using brain imaging. And finally, we will hear from the newest member of the faculty at Penn, Dorothy, who's a newly arrived and great knowledge professor in the law school and also the departments of philosophy, sociology, and Africana studies. And Dorothy is an expert on the ways in which sort of biologization of race, particularly in terms of genetics, but a lot of these lessons, I think, apply to neuroscience as well, have been used and keep it with the title Misused. So without further ado, I give you Janet.
I might try to pound the mic. Is that okay? Will that work okay? All right. I just am terrible with microphones for the most part. I hate those terrible squeaky noises and everything else. And heaven knows I don't want to distract from Gould and Morton. We try to do. I actually need to have my slide set put up. Okay. So, I do? No. Can you hear me? I'm not a big microphone speaker, and actually if I stand away from the mic, I can actually see the slide set a bit better. Am I really okay for most of you? Okay? All right. So I'd like to actually thank Mark. I'd like to thank the entire Center for Neuroscience and Ethics. It's a great pleasure to be here. I'm going to talk a bit about kind of a case study of bias in science that has to do directly with the Morton collection and later interpretation of the Morton collection. And I start with this slide because it is an especially famous mummy skull, which is part of the Morton collection. It is the skull from which Joseph Smith, the founder of the Mormon cult, is it a cult? No, it's a religion. The founder of the Morton religion, okay, I got it out, actually pulled the Book of Abraham. So it's a really interesting collection on many scores. And hopefully you'll all be able to get the opportunity to go down and visit an exhibit that we have on the first floor when you enter into the treasure part of the museum. It's an exhibit that my two co-curators are sitting in the room put together over the course of the last year. It's about race and the use piece of the collection and the modern understanding of basically what race is constructed upon. Okay? So we intend on doing a bigger exhibit on race at some point in the future, assuming that we live through the 105-degree temperature at this time. So we're actually adapting as we speak, just sort of sitting here. In any event, what I would like to do is basically give you, essentially this science story that involves Samuel Morton, a very famous Philadelphia physician. He is considered by many people to wear two different masks, depending on your particular viewpoint and what they actually mean. He is the father of scientific racism. He actually is the first person who tried to measure differences between people and use this and with more or less used in a way to the detriment of particular groups of people in the United States. And the second way that he oftentimes is referenced is as the father of physical anthropology, the field that actually is my primary field of study. Okay? What he did was he measured these skulls and came to particular conclusions. He actually ranked the different races in the collection based on cranial capacity as a proxy for brain size. And in doing so, basically did, as you would expect, given his background, given his agenda, rank Europeans at the top in this hierarchy, went to Asian people, from there went to Native American people, and then of course at the very bottom, people of African origin. So it's a predictable kind of tale, actually. So if we go through the collection, you're going to see a bit of the representations and how he actually made his measures. I must say at the onset that there is a little bit of kind of confusion associated with the way the slides are put together, because Morton measured cranial capacity by using the internal dimensions of the skull bones. And he did this in cubic inches. Nowadays, and people still do this, measure the inside of the cranial bones, and they do it in milliliters, so you know. Sometimes you'll see, rather than volume, information presented in weight in grams, and these are frequently used like alternatively in various kinds of parts of the literature. So know that it looks confusing, but they're actually sort of synonymous things. Okay? The notion is that brain weight and brain volume are about the same. At least this is the way it's been calculated based on certain kinds of experimental studies. This is Samuel Morton. Actually, this is a couple of the crania that actually comes from the cranial collection, which is here. He did amass about 1,200 in his lifetime. The collection goes up to about 2,000 crania because after his death, his student, a man by the name of Meigs, actually continued on with the collection. And he also, too, did the same techniques of analysis on the collection. So it really is a third again larger than it was in Morton's time. Morton also collected animal skulls, inferior animals, as he called them. He had approximately 800 of these or so. They are actually not retained as a collection at the Academy of Natural Sciences, and they don't know the whereabouts of many of them. But I say this because he was interested in doing interspecies comparisons, although he never comes forward and publishes on that particular topic area. The skulls are meticulously collected. They come from a variety of people from around the globe who sent him skulls. Usually these were other physicians, or they were people in the military who had access to these materials. Some of them are archaeological, some of them are living people. There are series within the context of the series that are very interesting. For example, there's an entire segment of series that are called the lunatic series. There's a whole series of idiots. These, of course, were people who were classified in particular ways. He was very interested in looking at size or abnormalities associated with the intracranial structures, so he could do this sort of physical identification of those variants. He well published the collection. I have to say that it's one of those sorts of things that oftentimes you're reconstructing things like this from notes. Actually, in his case, he was very forward and very upfront. He published two major volumes, Crania Americana, from which these two plates are taken. He published the second one, Crania Egypticana, And his purpose, interestingly, was said to be ethnological, okay? That is, or ethnographic. That is, he was attempting to not only just understand the shape of the cranium and measurements of the inside of the skull bones, but he was a person of his time who actually thought that the biological form of a person actually reflected their personality characteristics, including intelligence. Okay, so it was a whole parcel, basically, with this. He is, by anybody's measure or score, even using the frame of reference at his time, a flaming racist. Okay? I've had arguments with my own colleagues on a paper that he was not a racist, that they have made very, sort of, in my opinion, sort of crazy claims about this. He, for sure, and in his published work, okay, this comes out, but in his letters, it's even more blatant. He has, obviously, an agenda, and this agenda is going to come to fruition as he moves and looks at this collection. Interestingly, on his gravestone, the following words are present: okay, author of Crania Americana, wherever truth is loved or science honored, his name will be revered. And then in Latin it says, he does not die whose fame endures. When he died, his fame actually completely ended. And it's interesting that he or his family had a particular opinion of himself or of him, and that, of course, did not come to fruition. And there's many historic reasons why that's the case. And many people looking at this have said it's because of the racist views that he actually portrayed. But there's other historic events in the history of anthropology which probably put him in this particular position as the forgotten father of American physical anthropology. As all of the neuroscience people here can sort of well attest, there is this notion, of course, of the importance of the brain associated with intelligence, with mind, with the integration of sense modalities. And I just pulled this off of the website of the National Institute of Neurological Disorders and Stroke that give a very kind of good feel, essentially, of the thinking about the function of the brain. At the time of Morton, that kind of idea was also present, that this was the seat of intelligence, so his focus of study was actually on measuring the cranial capacity of the crania that he amassed as part of this collection. He comes from a school, essentially, of people who are called anatomical characterologists. These are people who think the form, not only of the cranium, but actually the whole body, is indeed associated with intellectual faculties, and these are innate, and that by looking at these bumps on the head, in this case I'm actually showing a phrenological view, or by size of the crania, that you could predict particular personality characteristics. And I'm sure some of you have seen the listing of characteristics that have gone along with many of these analyses. Europeans are noble, the most intelligent, and it can go on and on and on. I thought about reproducing one of these segments, but then I thought, well, it's probably pretty familiar to us all. I want also, too, to point out that there is a modern science of phrenology, actually. Not on many skinheads, I guess. And I did go on the web just because I was kind of interested in this, you know, thinking, I mean, where did this go and how did it die? Well, it is dead in terms of science, but it's not dead in terms of some segments of sort of popular interest. I also found, however, that there's a society of people who in their basements actually trephinate skulls, trephinate their own heads. That is, perform a type of surgery on the skulls. It is not called for medically, but it's thought to alleviate spirits in the brain and all these other kinds of things. So I guess there's always the person out there, and of course, you always see these phrenological shingles outside of various storefronts on the boardwalk in Atlantic City. So in other words, you know, it's well known as a pseudoscience, but there is still this sort of kind of common feeling, actually. And it sends tentacles into many parts of many people's lives, that bumps on the head, size of the skull, all of these things can be associated in some significant way with populations, with races, with individuals. Okay? It's not gone. The collection itself that we have on Morton actually does contain phrenological skulls. And I had a couple of interesting discussions with Michael Weisberg as to why Morton never conducted the science of phrenology, even though he has his medical degree from Edinburgh, which was the home place, essentially, of phrenology. He seems to have stepped down from the task, and it may be that he, as a very precise person, was not able to accurately render these bumps on the skull in a way that was useful to him in his analyses. So he leaves this aside. He, in Cranium Americana, has one of his colleagues, a very famous phrenologist by the name of Holm, actually do that chapter. I also should mention that at the time of Morton and for many years after Morton, it was quite common beyond measurements of the skull to use measurements of a variety of things in order to determine the intelligence, okay, of different individuals or even among species. So one of them is actually the measurement of what's called the facial angle. And again, you're going to see that the greater the angle, the more intelligent, okay, the higher on this natural scale, okay, the peoples that actually have that measurement actually are positioned. And that you can see, of course, again, reinforcing this notion of Africans at the bottom. Africans are all placed in a lot of this literature between orangutans and Europeans. Okay? So it's not just, skull measures. I also mention this because Morton collected two types of measurements. Actually, the thing called facial angle, okay, and the thing called cranial capacity. Those are his two areas. At the time, okay, recognition of prisoners, for example, was made through measurements. So there was in a sense, almost a blossoming into ways of using measurements for identification purposes. Identifications were primarily solidified within the prison system in the United States as a way of doing identity of individuals within the prison system. And of course, this was eclipsed later on at the beginning of the 20th century with fingerprints. Okay, so and they are randomly distributed. Body measurements were used also in terms of the identification of race. There was also, of course, the development and probably tooling on the part of Morton of a series of very particular kinds of measurement devices. So he became almost obsessed with accuracy in doing these measures. So when you actually sort of see the rendition of his work that I'm about to show you, he is amassing a collection of crania, heads. He is developing the tools and techniques to precisely measure them. And then he's doing an analysis of those measurements, and then he's coming to particular conclusions. He basically accurately measures everything. And like Martha is making a reference to, his bias allows a particular interpretation of those measurements. So it's not his measurements per se that are in error, it's his interpretations because of his viewpoint, his bias, and what he really thinks is associated with cranial capacity. He does produce in the catalog of skulls, man and the inferior animals, a very nice listing of each of the units that are composing his crania by population. So Swedes are at the very top. I apologize for the poor quality of this half of the slide. I apologize, I wasn't able to get kind of a better view of this. It was taken from a Xerox copy and it was available online. But you can generally get a sense that he portions populations in particular ways. And then at the very end, and very scientifically, he constructed a mean cranial capacity here done in cubic inches of each of the races. And of course, again, you're going to see that at the top, Europeans, I think it says 93.5, but I actually can't see it. I can't see if it's my eyesight or it's just so poorly represented there. And it comes from a variety of peoples in Northern Europe. And it's known that Northern European populations oftentimes have large cranial capacities in comparison to populations from other places on the planet. Morton's work was disputed and reanalyzed by a very famous evolutionary biologist by the name of Stephen Jay Gould. He began this analysis in '78. He further produced a volume, The Mismeasured Man, in 1981, and then he went on to do a second version of this, an expanded and updated edition in 1996. And one of the characters that is looming large in his work is Samuel Morton, and the bias that he brought to his collection, and the bias that he produced in his results. Morton, is a man who is known, very well known actually, as a person who was a champion against racism in all of its guises. Gould, I'm sorry, thank you. Oh my gosh, I have to go home. Gould in all of his guises. He determined, based on his analyses, that Morton's work was a patchwork of fudging and finagling in the clear interest of controlling a priori convictions. And actually how the story is going to play out is that me and my co-workers here at the Penn Museum and at the Anthropology department at Penn actually took a look at Gould. So we've got Morton, Gould looking at Morton, and then this generation coming in the 21st century looking at Gould. And all of us, reflecting on where this all went wrong, basically. And I know that there's many interpretations of this. I'm presenting, basically my ideas or my views, essentially how this transpired. So Gould, being a person who was a champion of the equality of races, especially when it came to all of these things associated with intelligence and critical capacity. He wrote an amazing, scathing group of critiques in the 1990s against the work Geoffrey and Mary called the Bell Curve. Some of you might have read those works, amazing in terms of the human which he brought to those sort of critical assessments. He takes Morton's work, and yes, thank you, I said Morton, right? He takes Morton's work and actually reassesses the numbers that are presented by Morton and presents a very different kind of assessed number of cranial capacity than did Morton. He also, basically, I think you can see, has a little bit of a seriation within this line of numbers. However, the discrepancy between low and high is indeed quite small in his calculation. So this brought up the interest in those of us here at Penn in going back and looking at Gould's work, because for those of us here at Penn, we recognize or realize that Gould had actually accurately measured all of the crania which were part of the collection. So a critique of the bell curve, just to give you an idea of where Gould is coming from. To make the ethnic difference argument work, there is a single general measure of mental ability, because of course, Gould is a great champion of moving against these simplifications associated with the complexity of the different mental capabilities of individuals, populations, and that IQ tests that purport to measure this ability are culturally biased. And the reason why I bring that up is because cranial capacity, now measured in with a variety of different tools, is oftentimes used as a proxy for intelligence. I mean, even today, even though it sounds like this shouldn't be the case, it is done even today. And when we actually went back and analyzed Gould's work, we found that Gould, actually was as guilty in a sense of bias, but in a more right-minded kind of position than was Morton himself. So he really fudges and finagles the data to basically show that there wasn't this sort of hierarchical kind of numeric chronology of human populations. And we were able to, after 10 years, actually get it published in PLOS Biology. It took us 10 10 years. Yeah, we were rejected every single place that we ever sent the manuscript. Okay. I find that interesting too. And we even had editors say to us that they didn't want to say anything against Stephen Jay Gould. Yeah. Isn't that interesting? And I was thinking to myself, I guess, I was sort of shocked. I think I still am a naive person. And I wouldn't think that a critique of a work was, you can't say anything bad against Stephen Jay Gould. I would think that it would have to do with the information which was presented, not in the person. Okay? And it's a shame because we essentially missed the opportunity to do something which a Nature editor actually put together right after the PLOS Biology paper came out, and that is the opportunity to discuss it with Stephen Jay Gould. Because, of course, wouldn't it have been nice to have this out in his lifetime rather than basically almost like sort of lobbying at bombs at a person who actually can't come back, okay, and say, essentially, what their thinking was when they did this reanalysis. And this highlights, I think, one of these other kind of components of science, and that is that critiques, okay, of work are not oftentimes accepted very well, especially when the critiques are very off-center. What happened ultimately in terms of our work, and really is devastating to all of us as a group, okay, it was actually hijacked by racists. Okay? Because if you say that these populations do fall into these numbers associated with cranial capacity, since they don't give up the idea that it's associated with intelligence and IQ and all of these things, in their usurping of this information, they see it as a vindication, okay, of their work. So we wind up being with all of these crazy, racist blogs, okay, including being the champion of one of the great racists that's still alive and is an academic and was working, okay, Philip Rushton, who some of you might have heard of. We're his champion. We're the guy's hero, okay, because, okay, we're basically not saying that these things, these, numeric determinations are associated with intelligence or IQ. He's making that leap. Our conclusion was, and rightfully so, that the cranial capacity, intelligence, and IQ is actually associated with, in the case of the way it's seriated in these populations, body size. Okay. So now you think, okay, and actually if you think back on that chart where the Morton information was presented at the very top, Europeans, if you actually, probably because of the bad nature of the Xerox, you probably didn't notice that listed at the top were the Dutchmen and the Swedes and the Slavs. Okay. These are today, and we're in the past, the largest people in terms of body height, okay, that have ever been recorded on Earth. Okay. And so, of course, they have larger brain sizes. Okay. Makes sense. The people that were the components of his African collection primarily came from West Africa, a Bantu group who in body size are quite small. So that explains that discrepancy. And it's easy to explain that discrepancy because, of course, this is the case, well recognized, well understood, the cranial capacity of women is also below men. And when you equalize for body size, they're equal to each other. Okay? In any event, okay, Martha sent me along this article, which I'd seen versions of, earlier versions of, but there is still, okay, the association between brain volume and intelligence. Okay? And this particular meta-analysis comes to the conclusion that about a third of the variation in intelligence can be associated with brain size. Okay? In this case, the brain size is measured not from cranial capacity, but through MRI studies, okay, of the internal, basically of the actual brain in living forms. Okay. I found it really interesting, of course, because the person also did not, okay, take into account anything about stature. There is no information about stature in the article at all. And so, of course, it's almost like a repeat of the same problem, but kind of like over and above a repeat, in the sense that this person should have been much more enlightened, okay, as to that association, and it is well represented in the literature, this association to body size. Okay? And of course, the issues with IQ and intelligence really come to the forefront in many discussions. Okay? I know Stephen J. Gould was talking about the social components associated with this, and I love this slide, okay, and I think you can all see it right away, okay. That, okay, it seems as if tall people who probably have big brains are very socially competent. You see what I mean? So part of that measure, okay, of IQ is associated with cultural competence. So you can see the position of American presidents, okay, vis-a-vis the average at the present time of male height, okay, at 5'9.5" inches. This is actually the average measure as we can project it back into the past. So these presidents also, too, are above the average, okay? And so all of these presidents, okay, basically with their tall body height, obviously are at some kind of advantage in a sense, okay, when it comes to social competence and probably in terms of measurements of intelligence, okay? We also, too, have to make a quick mention here, and then I know I have to keep quiet because you have got two wonderful speakers coming up, that many, and this is through the 20th century, many famous people donated their brains to science to be measured, because this idea of measurement and intelligence always sorts of, lifts its ugly head. And my favorite story, of course, has to do with Einstein's brain, okay? The average human brain size is 1,400 grams, okay? So this is where I'm making this transfer into weight, and as I said, that they're going to use that also to almost synonymous with volume. And Einstein's brain is actually one standard deviation below the mean. Okay? So now what happens to Einstein's brain? It's actually something which is part and parcel of our understanding of the brain and brain evolution, and that is, it isn't about size. Okay? It isn't about most things, actually. It's hard to compartmentalize in particular ways. But because he's Einstein, okay, they're going to find a way out of this problem, okay? So with Einstein, of course, what they do, and this is actually somewhat right-minded, at least on a gross level, his brain was left, okay, to science. Sections were made. In 1999, a paleoneurologist by the name of Dean Falk at the University of Florida did a section-by-section analysis of Einstein's brain and found that he had a disproportionately large area in a segment of his parietal lobe. Okay. She also came to the conclusion that there was no lateral sulcus on the cerebral cortex of his brain, thinking that this made there was a different kind of a connectivity in that area of his brain, and of course then reduced Einstein's brilliance to all of these topical, actually some of them are internal, but here the topical differences between this and what would be kind of a normal or average kind of brain. We do, in my particular field of study, because I do human evolution primarily, look at, not obviously brains, we don't really have those when it comes to fossilized forms, but we do look at the overall compartment of the cranial cavity in order to get a sense of the differences between a brain of a human and a brain of something like a chimpanzee, and lots of the differences are actually organizational. So if you took a chimpanzee brain that is at about 400 milliliters or so, and expanded it to 1,400 milliliters or so, it still wouldn't have the behavioral components of a human. There's other parts of this which are part and parcel of humanness. And then, of course, many people have noted, although not available to Morton at this time, that brain size always has to be in correspondence to body size. And then you can see this, it's produced here as the encephalization quotient, but you can also see it produced as brain size as a percentage of body mass. And if you were talking about an encephalization quotient, then humans do indeed have quite a large brain. They can equalize for body size. But if it's by percentage of body mass, then a mouse is truly astonishingly brilliant. I won't go on the details of this slide. It actually has a couple components to it. But it actually is the correlation between brain size and body size, so you can see it across a whole great group of vertebrates. Okay? And modern humans are up at the very top here. Okay? And of course, sort of distorting this the structure essentially of higher vertebrates. I don't know what exactly how you would say that shape is, okay, but distorts that. Okay? In other words, we are a little brainy, okay, in proportion to our body weight. And we do apply this oftentimes to many studies in human evolution. And my final slide, just because I think it's kind of fun, and we don't necessarily talk about this all that often, but when we actually look at brain size over the course of human evolution, it appears as if our biggest brain, okay, members of our lineage are actually Neanderthals, okay? And in the last 100,000 years, brain size has actually reduced, okay? And you know, basically, how do we understand something like that? And of course, we'll go on and we'll talk about body size of Neanderthals. But in a sense, okay, you'll see all kinds of ways of explaining that away too, because we have to be fast. It's like a species, okay, kind of racism, you know what I mean? And the racism in this case will frequently cast Neanderthals as bumbling idiots, for example, even though they've got these massive brains on their shoulders. Sometimes my colleague who now teaches up at Princeton, but who was here at Penn for a long time, says that it's okay to be prejudiced against Neanderthals because none of them can come forward and start. So thank you very much, and I'm sorry I went a little bit.
So I presume we're going to do questions at the end. Great. Could someone please switch the slide deck over to skulls to scan? Well, good afternoon, everybody. So I apologize, I think as the prior speakers did, for the heat in the room, but I will try and move briskly and be animated, so perhaps try and take your mind off. So, well, that was really a fascinating conversation, a tough act to follow. I'm going to, in the time I have, talk a little bit about modern brain imaging techniques, and as they've been applied to study of group differences. In particular, I want to focus on two types of error that turn up in these sorts of studies. And I think what you'll see is that these types of error are going to be quite familiar from Janet's talk as well, the types of errors that have been suspected previously. So let me just begin by saying that, right? So let me begin by saying I don't study racial differences. I'm a vision neuroscientist. I do most of my work in the back of the brain, looking at the coding of visual information. And further, I don't find studies of group differences in general to be particularly interesting or in many cases very well founded from the get-go. Nonetheless, actually, I think they are interesting as an example of scientists who wish to project their beliefs upon researchers. So while science is a method to discover truths about the world, scientists, of course, are human beings, and they're subject to biases and stubbornness and the general ability to ignore evidence that would contradict a cherished hypothesis. Well, let me lay my cards on the table. I think despite the biases and errors that I'm going to describe, I do think that science is a cumulative process. I think we do get better at making accurate measures of the world and predicting the properties of the physical and biological universe. And that applies not just for simple instrumentation like microscopes, but to more complicated things like brain imaging. So I think in the last 150 years, there's been a dramatic advance in the kinds of things that we can measure about neural anatomy and function. And we've moved from early measurements of brain, of skull size and shape and capacity into some enormously subtle and powerful measures of brain structure and function. And those are the measures that we're going to talk about today. So in this talk, I'm going to discuss the use of these sorts of modern, powerful brain imaging measures to examine differences between groups of people. And because the topic today is about race, that's the studies that I'm going to highlight. I'm going to use examples of imaging studies, which I have 3 that we're going to work our way through, which have been designed to identify racial differences. So as you might imagine, brain studies of racial differences are not a particularly active area of scientific research these days. You can imagine different reasons why that might be. But the topics I'm going to discuss today are equally applicable to other sorts of studies, other kinds of group differences. And in particular, I'd like to highlight the studies that have been conducted looking at differences in gender and sex and sexual orientation. There's a huge literature of this stuff, and it's been going on for decades. And I think some of the same critiques that I'm going to offer today about studies of racial differences apply to this literature as well. And if you're interested in a very good read on this very topic, I recommend to you this book by Cornelia Fine called Delusions of Gender. It's really a great book, and it basically takes the same sorts of topics that we're going to be talking about today and applies them to this industry of neurological studies of gender differences. So something a little homework reading for later. All right, well, in this talk I'd really like to go through two sorts of bias or error that can creep into neurological studies. So the first of these is measurement itself, right? So while it's possible that a scientist is just a simple fraud in making up measurements, I think a much greater problem, and the problem that, in fact, Morton was accused of by Google, is that there's bias in the measurements that are obtained. Now, in the modern context, I'm going to argue that actually, while we have very precise and accurate ways of making measurements about brain structure and function, there's a way for bias to creep into measurement simply because we have so many ways of making those measurements. There's so many different things that you can measure that if you've got a bias or a real interest in finding a difference between a pair of groups, you're going to be able to find something. So that's one type of bias we're going to talk about. The second type is of interpretation. Again, this is a topic that came up in Penn's talk. Once you've actually found a difference between two groups, in this case a brain structure feature, what kind of interpretation do you apply to it? And that's really where the tricky part of work begins. So the studies I'm going to describe today all use MRI imaging, which is a technique that probably many of you are familiar with, probably many in the room have been inside a clinical MRI scanner. This is a method that uses a combination of magnetic fields and radio waves to measure non-invasively images of the human brain while people are still alive and healthy and using their brains. It doesn't require any harvesting of skulls or anything else. This is just something you can do on volunteers in a non-invasive manner. I'm not going to say too much more about neuroimaging and MRI as a technique, though we are going to get into some of the nitty-gritty details of the sorts of measurements that one can make with it. Okay, so now the MRI scanner can be used to produce a vast array of measures of the brain, and I'd like to orient you to some of these pictures that we're going to see. So mostly we're going to be looking at horizontal slices. So this is a side view, obviously, of someone's head, and the red line indicates where a horizontal slice might pass through the head. And so we're going to be looking at what are then called horizontal images of the brain. And so here's one example of one of those images. You can run the scanner in such a way as to obtain what's called a structural image of the brain, which is ideally suited for making measurements about brain anatomy and brain structure. Now, in contrast, you can run the scanner a different way and come up with what are called functional images of the brain. And these images can be used to measure ongoing neural activity. In fact, these sorts of pictures are the basis of studies that then look at how different emotional or mental states or intellectual states are associated with changes in ongoing neural activity within the brain. This is a fascinating area of study, and also one in which group differences have been sought, but it's not a topic that I'm going to focus on today. So we're not going to hear too much more about functional images, but instead we're going to focus on, in fact, all the things that one can get just from a structural image. Okay, so, back in Morton's time, as you've heard, there were several measures that one could get. You could measure aspects of the shape of the skull and from different angles, and you could also measure something about skull volume, or brain volume. Now, in contrast, in modern imaging techniques, you have many, many more sorts of things you can measure. I want to give you a little sample of what some of that variety is like. So, okay, here's just a standard anatomical image of the brain, and there's lots of different sorts of information that can be pulled out from an image like this. Just for example, you could simply identify where the brain is located within the skull and obtain a measure of brain volume, just as has been done with other techniques going back centuries. But that's really only the beginning. So another measure you can get is sort of a regional brain volume. So now you say, well, I'm going to be interested in measuring just the volume of the frontal lobe, or just the volume of the parietal lobe, or the corpus callosum that connects the two hemispheres. So there are these different measures that you can get. There's yet another way you can run the scanner to measure sort of white matter connections. So the brain is composed of different tissue types. The white matter are sort of the telephone cable connections between different regions of the brain. The gray matter, those are where the nerve cells themselves are located out on the covering of the brain. So there's a sort of technique that you can use these MRI scanners for to measure the quantity and number and position of the connections between different brain regions. But going on, we can measure something called gray matter thickness. So it's possible in these images to identify what's called the cortical ribbon, this thin 2 to 3 millimeter layer of gray matter, the cell bodies of the nerve cells themselves that are that cover the surface of the brain, that make up the surface of the brain. So you can measure that and then measure the thickness of those portions. And actually, once you've measured gray matter thickness, well then you're off to the races, because it turns out after you identify this gray matter sheet, there's a whole new set of things you can measure. And the reason is because that gray matter thickness allows you to treat the brain as a single topological sheet, a single continuous surface. And in the same way you can take a ball of paper and unscrunch it and flatten it out, you can do the same thing with the surface of the brain using some of these digital techniques. And so now, once you've got this sort of surface representation and measure the curvature and folds of the brain, well, now there's more things to measure. So now you can take a look at the folding pattern. It's something called gyrification, which is the relative amount of folding that you have in different people. And you can look at things like hemispheric symmetry, how similar the two hemispheres are, on and on and on. So and this is just the beginning of a huge list of different sorts of objective anatomical measurements that you can make using these sorts of imaging techniques. So now, what I'm going to argue is that in addition to these different things that you can measure, these different measures that you can obtain, there's yet another dimension entirely that makes this problem even more vastly complicated. And that is that the brain's a big place, okay? So, the brain is a volumetric structure, and we make these measurements over lots and lots of little dots that make up the images that we obtain. Those things are called voxels that compose the entire volume of the brain. And a typical study might have 200,000 of these voxels, right? So now what you can imagine doing is comparing each and every one of those measures at each and every one of those voxels, and now looking for some kind of difference. Well, it turns out that if you have 10 measurements and 200,000 different places to measure it, there's a lot of different ways you can get fooled, right? There's a lot of different ways you can find some random fluctuations, a little bit of noise in your measurement. So even with a very precise and modern imaging technique, there's still going to be a little bit of noise in that measurement. And if you go look at enough different places, eventually you're going to find some kind of difference. So this is the first kind of error that I'd like to try and illustrate the bias that can creep into these sorts of studies. And that is biased reporting of positive findings. If you are predisposed to believe that there must be some difference between two populations that you're studying, and if you look at enough measures and enough locations, you will find something. Right? You will be able to find something. And that really is the core limitation of some of these studies. Just to illustrate one of these studies that I came across, here is a paper from 2010. And in it, the authors measured the volume of different brain regions from, as they put it, Caucasians and ethnic minorities. So they claimed that African Americans exhibited a larger left in the OFC means orbitofrontal cortex volume. Okay, so the African American group had a larger size of this one brain region, which is illustrated in red on that side view of the brain. And they found actually that it was a pretty significant difference, right? So when you do these sorts of measurements, you can apply a statistical test and come up with a probability that you were fooled by noise. How likely was it that this thing you measured, this difference between the African American and the Caucasian group, was the result of this random noise? And in this case, they said it was P equals 0.008. So what does that mean? That means that only 1 in 250 times would you be fooled by random noise into believing that there was a difference when there really wasn't a difference. Well, wow, that sounds pretty impressive, right? Except, here's where we get into this problem with lots of measures and lots of places to look at it. So problem number one, they had a fairly small sample size. They had 44 Caucasians and 33 ethnic minorities, of whom only a subset were actually African American. And of that group, it turns out that they weren't even age matched. So they had to take a subsample of the subsamples to get two groups that actually had the same ages. If you have small sample sizes, then boy, you are looking for an opportunity to get fooled by noise. Then here's the next problem. Did they go into this study assuming or with the basic hypothesis that the orbitofrontal cortex was going to be different between the two groups? Nah. They went looking for a whole bunch of places. They looked in the amygdala and the hippocampus, the lateral ventricles, and the caudate nucleus. They took all these different measures and they measured it on both sides of the brain. And by the time they were all done, they were testing 11 different regions of the brain to see if there's a difference between these two groups. Well, if you're willing to accept just a 1 in 20 chance of being fooled by noise, that's the scientific standard, and you look at 11 different regions, then lo and behold, you got a 43% chance of finding something just by random dump luck. Okay? So many sorts of studies of group differences. So this is a problem that exists in all of science, not just in studies of group differences. But again, if you're predisposed to find a difference, you can often find one. Okay, so in summary, I think that this kind of error, this sort of issue, is really one of a fishing expedition. If you fish long enough, if you put your line in the water long enough, you'll catch something, even if it's just a tiny. Okay? So I don't want this critique, though, to necessarily mean that it would be impossible, in principle, to find some kind of measure which is reliable and completely different in some brain anatomical feature between two groups, right? So it is possible to do studies in such a way that you can test a hypothesis, replicate it, and confirm that there really is a difference. Okay? And so actually I'd like to illustrate what I would hope to argue is an uncontroversial measure of racial difference in brain. So let's see if I can give this a shot. So before we even get into group differences, let me just start with the observation that in brain imaging studies, it is often important to collect data, or it is often a goal of the study, to collect data across individuals, right? You would like to try and make a claim about what's true about a group of people. Right? And to do so, then, you have to grapple with a fundamental problem, which is that different people have different shaped brains, right? And forgetting, we're not into group differences here. This is not about gender differences or race differences. But a group of people from a homogeneous population are all going to have slightly different shaped and sized brains. So if you'd like to try and take this group of people and then combine their data in some way to say what's in common across the group or what's different in different individuals, you need a way of solving this problem. And a standard technique that's used with people is to normalize that data to a common atlas space. Right? So you create an atlas, a template, which represents the central tendency of the group of people you're studying. And you normalize all those brains into this common space. And so then you now have a common reference frame in which to compare data across different people. But different groups of people might need different common atlases, right? So just for example, it's necessary to create different atlases for children and for adults. So if you're doing a study in children, children have different shape and size brains, they have different sorts of myelination of the white matter. You need a different template to study that group versus another. All right, well, it turns out that when neuroimaging was just arriving in China, and this was becoming a very active area of research within Chinese academic centers, investigators there felt that they needed their own atlas, their own common template space, in which to normalize the data that they collected. Okay? And I should actually point out one little subtlety here. The title of the paper is that it's the construction of a Chinese MRI brain atlas, a comparison between Chinese and Caucasian cohorts. You know, a little bit of a nuance there. It's not just Caucasians, it's Canadians. So it turns out that the standard atlas that's used in this field is derived from a group of students at the Montreal Neurology Institute. So that it's actually, so we're going to be talking about a group of Chinese participants versus Canadian participants. But anyway, it's just a property of these two populations that they study that there is actually a difference in brain shape between the two groups. Okay? And this is, you know, as laid out in this image, just putting side by side the central tendency of the two populations they study. Okay? So this is actually a very important methodological observation, right? So if you want to do studies of different sorts of populations, you need to know something about the central tendency of the population to do this normalization, then, okay, you need to know this. The trouble then becomes, well, trying to apply some kind of meaning to this, right? So in this next and final step, where you take this difference and now try and assign it some kind of interpretation, that's when the bias comes back in, right? And it's really something like this. So, you know, you've got this image, and you want to try and know, what does this difference mean? Right? So if that's your goal, you found a group difference, now you actually want to ask a question, what does it mean? And that's where the final problem arises. And the problem is, I think, because these brain images serve a little bit like a Rorschach blot, right? It is, I would argue that these images allow the viewer, the scientist, to sort of project upon the image their own interpretation of what the group membership means. And this is particularly problematic as group differences are not subject to experimental control, right? What I mean by that is I can't somehow create a group of 20 Canadians who do not nonetheless have some particular Canadian-ish property that I think is essential to the explanation, right? So here's a group of people, they're Canadian, yet they don't have the one thing that I want to call is the critical Canadian-ish thing that makes their brain different from other people, okay? And that's the core problem. So here's just a final example, a final study. in which a group difference was sought and then an interpretation applied. Okay? So this paper was published in 2011. It examined differences in that gray matter cortical thickness vector between a group of East Asians and Westerners. Okay? Specifically, they studied a group of volunteers in Singapore and a group of people in Chicago. Okay? And they found that the group from Singapore had a slightly thicker gray matter layer, that's what the red stuff is, as compared to the group from Chicago in this area of the frontal lobe. Okay, that was the finding. Well, then here comes the trouble. What does this mean, right? What interpretation would the authors like to apply to these images? Well, here's what they came up with. East Asians and Westerners differ in cognitive processes because of cultural biases to process information holistically, East Asians, or analytically, Westerners. Okay? And the finding of thicker polymodal association cortex, that was the red block, in the U.S. young folks would be consistent with such a framework, right? So their interpretations, the way they interpreted these results, was that what the key difference is this cognitive difference, that you can take a look at these two populations and allegedly there's a difference in the way that folks from these two cultures work with information. Some process information in a holistic manner, integrate all the parts into an integrated framework, whereas the other group is allegedly more analytical and processes each piece individually. Well, gee, gosh, can you think of any other way that a group of people living in two different cities on opposite sides of the globe might be different? Well, I mean, here's some, right? I just take these out. But of course, more importantly, I have no idea, right? And neither do the authors, right? There's no way of knowing which particular property of difference between two populations is actually responsible for this thing, okay? So in the end, I think that this is the final form of bias, which is the most critical and problematic of these sorts of group studies. And that is when faced with a brain image that can mean almost anything, right? The interpretation that the authors supply tells us much more about what the authors believe about the group differences than what the image actually tells us. Okay, I'm going to stop there. Thanks for your attention.
Exciting slides because my role here is to be a commentator, and so I wanted to hear what the speaker said and not prepare something in advance. And I think what both Janet and Jeff show is what Martha began with, the fact that science operates in a social context, and scientists are not immune from the biases in society. Even though they may think of themselves as being in a hermetically sealed lab that doesn't have any effect from the broader society. I think what one of the most important aspects of the social context that scientists operate in is inequality, social inequality. And one of the main axes of social inequality in the United States and other parts of the world is racial inequality. And so I don't think we should be surprised that when scientists measure the human body, they are affected by racial inequality that exists, and that their findings have been used to support that racial inequality. More deeply, I think, the power of neuroscience to shape the way we think about racial inequality and to be shaped by that is that it gives biological explanations for social differences. And in particular, it provides a biological definition of race. And those two aspects of the relationship between biology and society are extremely powerful. And if we can explain social inequalities, why it is that blacks, for example, are less wealthy, more likely to be incarcerated, do less well on intelligence tests, and with a biological explanation, then the inequality seems to work. If we can believe that these social categories called races are natural biological categories, it's more easy to accept the biological explanations for inequality among these groups. So Janet showed very clearly this long legacy of scientific racism. And she said that Samuel Morton was the father of scientific racism because he was the first to measure racial differences in a scientific way. Which is true, but I think we should remember that there is an even longer history of scientists identifying racial difference and defining race as a biological category. So we can go back to Carl Linnaeus, who in the late 18th century developed a classification of races, and lots of other European naturalists who developed typologies of races. They didn't have the instruments that Samuel Morton did, so they looked at things like skin color and hair texture and that sort of thing to say that this showed that there were natural, innate differences between the races and that you could divide up human beings into races. What Morton did was measure it using the most accurate tools of his time. And so scientists went from just identifying these outward indicia of racial difference to digging a little bit deeper, going into the brain or other parts of the anatomy to prove racial difference, and then conclude, as the European naturalists before them did, that Europeans were superior to other groups and Africans were at the bottom, closer to apes. I hope I don't have to mention that all this is going on at the time of European enslavement of Africans and colonization of others around the world. And so it's not just an accident that they happened to come up with this. They came up with typologies and explanations that supported European slavery, enslavement of Africans, and conquest of other peoples around the world. And so now we're digging even deeper. Scientists are using more accurate measures. Remember, during the time of Samuel Morton, you may now say, laughing even, at his types of measurements. But at the time, he was revered for using the best tools available. And similarly today, many of the scientists who continue to look for racial differences, but now deeper in the body, are revered for being brilliant scientists using the most cutting-edge technologies. My work recently, which I published in a book called Fatal Invention, How Science, Politics, and Big Business Recreate Race in the 21st Century, looks at how scientists are digging even deeper now to the molecular level to find in genes racial differences. And the same kinds of studies that Jeff showed about differences in the brain using neuroscience, there are dozens of studies being published that claim to show differences among races based on genes. Or another way of saying it is that race exists at the genetic level. Now, we might call Samuel Morton a flaming racist, his work pseudoscience. But again, at the time he was producing it, it was considered top-notch science. And scientists today who continue to look for racial differences in the brain or in the genes and define races as natural categories that are biologically identifiable, which is something I thought might not have come out in the prior talks, that we should also contest the view that not just that these natural races have these differences, but that the very idea that human beings are divided into a handful of races that are biologically distinct from each other and that cause the differences we see in welfare of people in society, that very concept that scientists have supported for centuries should be subject to scrutiny. Okay, so Morphy is a flaming racist and pseudo-scientist. Now, today, scientists looking for racial differences at the molecular level would say that, no, but today we're using more accurate technologies. So we're not like Samuel Morton because we're using fancy algorithms and we're using brain scans and we're using huge DNA databases and we're using computer science to find these differences. So we're different from Samuel Morton, and they would also say we're not racist, which is probably true for most of them. We're just, we don't think people are unequal, but we do think they're naturally different based on race. I would argue that the very concept that human beings are divided into 4 or 5 groups that are biologically distinct from each other, the very practice of doing that and trying to find ways in the body to distinguish between them, that very practice is a misunderstanding of human genetic diversity and commonality. So the errors that Jeff pointed out in some of these neuroscience studies, you can find very similar errors in many of the studies looking for racial differences at the molecular level. For one thing, in many labs, the DNA comes into the lab already grouped by race, as if, so there's already an assumption that there are differences based on race in the very material scientists are using. That's not true for all labs, but in some, they are. Now, how are the races identified? Usually, in scientific research, it's self-identification. So it's just what the donor said he or she was. And that could vary. My race could vary according to what I say it is, or in different countries I might be considered a different race than what I'm considered in the United States. Then there's the problem of residual confounding, because very often scientists will control for one small social variable, and if they continue to see a racial difference, leads to the conclusion it must be genetic. So a study, for example, that's trying to discover why it is that blacks are more likely to have, black women are more likely to give birth to preterm babies than white women in this country, controlled for welfare receipt. And when there was still a difference, just left to the conclusion, well, it must be a genetic reason for this, when there's so many other social causes that might create the difference. And then also, I thought it was interesting, Jeff, your point about all the different ways of measuring the brain. Similarly, there's so many different ways of collecting DNA data. Do you go to the most isolated indigenous tribes around the world, or do you collect from a big urban center? You're going to get a difference in your results that way. Do you group all white people together, or do you make a distinction between Canadians and white Canadians and white Americans? Why would you group some people by nationality and some people by race in the same study? That goes on all the time. Study that determined who was Hispanic by looking at the participants' last names. Things like this where there's so many ways of deciding what race people belong to and how you're going to use it, and then how many races there are. So a very prominent study that many people interpreted as showing that human beings are divided into 5 or 6 races, but it turns out that if you tell the computer to find 20 genetically similar clusters, it'll find 20. Tell it to find 100, it'll find 100. So the number 6, right, conveniently mapped onto our current conception of the number of races. But we could say that in Africa alone, if we wanted to, we could divide groups into 1,000 races just in Africa. So these tests depend on what people are looking for to begin with, how you define the categories you're using, and if the scientists are really willing to look for possible social explanations, which I would argue are much more plausible than genetic explanations. It's much more plausible to find a social explanation for why social groups have such different outcomes in this country than to argue it's because of their genes. So I'll just end by saying that I hope that there can be more collaborations like those at the Center for Neuroscience and Society, where people from various disciplines, anthropologists and neuroscientists, geneticists, sociologists, legal scholars, can come together and think about better ways of studying human beings than relying on the same categories that Carl Linnaeus came up with in the late 1700s. You know, why do we do that? Can't we advance science more? And I like when you said, on balance, science progresses, it advances. I think racial bias has been a huge impediment to scientists' contribution to our understanding of human beings, including our brains, our genes, and why we behave the way we do. And so that's my plea, that we can come together and come up with a way that truly revolutionizes the way science thinks about human commonality and diversity, so we aren't really doing what Samuel Morton did in a fancier form. Thanks.
work is considered to be central in modern physical anthropology, and moving almost completely away from this notion of a skull associated with race. There are other people, however, also anthropologists, who have, coming from a solidly different direction, have come to very nicely quantifying, again, you're talking about instrumentation and things of this nature in a much more sophisticated way, including through CT scans. So we CT scan all of our collections here, and have used our CT scans to sort of talk about race differences in the skull, not just by a single measure like a facial angle or by cranial capacity, but by a whole host of other dimensional characteristics. So, what was the bottom line of the question?
I guess both, Boaz and Wolf, Mom also said, I think they all
So you think, yeah, I mean, they definitely had their own sort of set of biases, and there's always a whole other set of critiques. I don't know that I feel that we move forward in the study of crania associated with race, and it's mostly because it's not something which is universally accepted. Well, I should actually qualify this for just a minute so you can make it make a bit more sense. A lot of the craniology that you see in the United States today, and there's quite a bit of it, and it is associated with race and race categories, is associated with the use of cranial measurements for forensic purposes. So what happens is, on the one hand, you've got people scientists who are feeling like, for legal reasons, and for important legal reasons, for identification reasons, have really gone back to very solidly trying to identify characteristics that are associated with race. There's a wholly different rationale for doing so. And those people, basically will tell you that there are race differences in the skull. And they have databases that basically enforce this particular viewpoint. A major one is distributed, you can purchase it, police departments have it, all over the world. It's called Fordism. So it depends on sort of your part of the field and your thinking and how this is achieved and how it's moving forward. Did Gould impact positively this notion of craniometrics associated with race? I would have to say yes, if the review process is any indication. Boaz still stands very strong within anthropology and is frequently used as a model for moving away from these typological methods. So yes, we have moved forward in some capacity. So when it comes back to the practicalities of everyday life, we still go back to the typology, and that's which one is it?
So what you asked me that question directly, I would say that first of all, I think it's not the case that I would put that the idea that race is a political social category has completely overwhelmed science, and no one believes anymore that race is a biological category that can be measured genetically. I was surprised about it. I thought that we had gotten to that point until I started doing research for my book and discovered that no, there's lots and lots of studies being published and funded whose purpose is to find racial differences in the genes. So I don't think it's accurate to say are we swinging, one way or the other, because I don't think things have changed as much as many of us might have thought. But I would also say that even though there's, I sort of premise that we all have biases, and it's not true that science is immune from bias. No scientist is. So I would dispute you that scientists who are left-leaning are influenced they just say we grouped. We had 33 African Americans and 40 Caucasians. They never define what is Caucasian saying as a white person. Does it matter if this Caucasian came from Canada or the United States? Does it, how do you define these groups? And no recognition that these are social groups. Just basically Black or African American in the United States means anyone with any African ancestry. That's a social definition. That it's not commensurate with how we define white people or other groups. So we could just as easily define, except it wouldn't be politically possible, but we could have been that whites were defined as anyone with any ancestry. Well, then all of a sudden, I'm white, right? So why? Not because my genes changed, but because of this social grouping. So when genetic researchers are using the category Black and white, the Blacks in their study may be more genetically, some of them, more genetically similar to the whites. They may have more European ancestry than some of the other Blacks in the study. It's imprecise. And then the other point is that if you're studying a social category, you shouldn't treat it as a biological variable. That's not to say it can't have biological effects, but the category itself is treated as a biological variable. And then that then biases the study against finding the social causes for what you're trying to understand. And that's a big flaw in the study, because it could be that the social causes are the better explanation, a stronger explanation, and which then has a big impact on how we're going to address what we're probably looking at. Are we going to address it by making changes in society, or are we going to address it by reducing film, for example, that's geared toward particular genetic makeup? So I think that I wouldn't say it was the scientists who said, I'm not going to touch this because it's too controversial. I think we need to look into this controversy and figure out a better way to approach it. I would say a more scientific way to approach these questions that is being done right now.
I don't know if this is a biological or social piece of data, but I